Dear colleagues, as we unfortunately have canceled this year's World Congress in Montreal, we thought it would be a good idea to at least share some highlights from the virtual Congress that took place back in February. Before the video starts, I would like to announce that we will open the Symposia submission website in July again for the 2022 World Congress in Taipei, Taiwan. I look forward to seeing you there in person next year. And now please enjoy the video. Oh, and don't forget to subscribe to our YouTube channel below for free so that you can watch more video in the coming weeks. Thank you.
Welcome everyone to this lecture from genes to clinical phenotype in ADHD across the lifespan. My name is Barbara Franke. I'm a professor of molecular psychiatry at Radboud University in Nijmegen in the Netherlands. ADHD is a multifactorial or complex disorder, meaning that both genes and environmental factors are involved in causing ADHD. And if we look at a group of individuals, then in each of these individuals, multiple genetic variants and environmental factors are involved in causing the disorder or are involved in causing the disorder. And if we look at the entire group, then in different individuals, different combinations of genetic and environmental factors can play a role. And this makes it so hard, this heterogeneity makes it so hard to identify the genetic factors that are involved in ADHD. So back to this, very important seminal study published in 2019, the GWAS meta-analysis for ADHD in more than 20,000 cases and more than 35,000 controls. What we can extract from such genome-wide association studies are, of course, the individual genetic variants and genes that are associated with the disorder. And we can use those to identify biological pathways that play a role in the disorder. But we also can extract heritability estimates based on the common genetic variants. So which percentage of the total heritability are explained by this common genetic variants. We can perform genetic correlation analyses and estimate genetic correlation here as indicated as RG with other disorders and traits to tell us about the relationship between these disorders and traits. And we can derive polygenic risk scores for each individual to tell us about the risk that an individual carries for this disorder of phenotype. So as for heritability, we showed that the common genetic variants explained about 21% of the heritability of ADHD. So as I said, about the total heritability of ADHD as from twin studies is about 74%, and more than 20% of that is explained by the common genetic variants. So we've covered quite a bit of ground, and the last few minutes I want to take to look into pathways that go from gene to disorder. And if we look at the genetic contribution to ADHD, I'm always using this pyramid on its head to explain the pathways from gene to disease. So the genes up here are contributing to biochemical processes that support cell functions, and these cell functions are involved in shaping the morphology as well as the function of individual brain units. And together, this functioning and morphology of the brain actually leads to behavior and to disease symptoms. This is a convergence model where we have a lot of genetic factors up here. And in each step, probably there is a convergence towards each other, meaning that several of the genetic factors contribute to an individual, a single biochemical process, and several biochemical processes contribute to an individual cell function, etc., etc. There's of course not only genetics involved here, there's a lot of environmental influences across the entire pyramid, so at all levels probably environmental factors also are involved. So genetic factors lead to disease symptoms by going through these different levels, and we can perform research at all these different levels to understand what is going on. And this actually has to be interdisciplinary research, which we do, for example, on ADHD in the Psychiatric Genomics Consortium, in IMPACT, as well as the CINP network on ADHD across the lifespan. And we do that in many different consortia that are involved in the research of ADHD. In my own lab, we have developed a whole battery of different assays and different approaches to be able to look at these different levels of analysis. We use bioinformatics to understand biochemical processes and cell function in some cases. We use induced neurons to look at cell morphology, at cell functions. We use animal models to look at the context of cells in the brain. And we also use cognomics or imaging genetics to directly couple genetic findings to brain phenotypes in vivo. So as for take-home messages, the biological processes altered in ADHD include those for neuronal development and differentiation, as well as neuronal functioning. And looking forward at what these findings might mean for future clinical management of people with ADHD, it could help us to develop diagnostic biomarkers as well as novel treatment targets for ADHD. Then the genetic contributions to persistent ADHD in adults and childhood ADHD overlap strongly but not completely. And given that non-complete overlap, there may be room for the development of prognostic biomarkers if we can find those genetic variants that differ. Then the finding that the same genetic factors contribute to ADHD symptoms in the general population and to clinically diagnose ADHD. It could help us to have measures of genetic burden based on polygenic risk scores for each individual. And that could help in early diagnosis. And also it could make us rethink the diagnostic thresholds and age of onset thresholds that we're currently using in the clinic, and perhaps question whether these should be applied very strictly. And then the finding of limited specificity in the genetic contributions to ADHD, so much genetic overlap with other psychiatric disorders, as well as with non-psychiatric comorbid disorders. This could help us to personalize therapy and to develop prognostic biomarkers, but it also more generally could help us rethink the psychiatric nosology, knowing that there is so much overlap between the different disorders, and questioning whether, based on biology, we could think of a better categorization or better specification of disorders more generally.